Biomolecules. The chemicals or molecules present in the living organisms are known as biomolecules. The chemicals or molecules which are present inside a living organism living organism is called biomolecules. Okay, then cellular pool. What is cellular pool? The collection or sum total or sum total amount of biomolecule biomolecules, compounds and ions present in a cell is called cellular pool. Okay, the collection or sum total amount of biomolecules, compounds and ions present in a cell is called cellular pool. Then types of biomolecule. Okay, which type of biomolecules are present inside a organism. Then at first we can classify the biomolecule into two groups that are these are inorganic biomolecules and organic biomolecules. What is inorganic constituent? The inorganic constituent, the substances which are present in a cellular pool, means the minerals, gases and water. Okay, which substances are inorganic? Inorganic substances do not contain carbon along with hydrogen. Inorganic substances are those which do not contain carbon along with hydrogen. So these are the inorganic substance. What is organic substance? Which possess carbon and hydrogen. hydrogen. These are called organic biomolecules. As example, the inorganic biomolecules, of a cellular pool are minerals, water, and gases. And the example of some organic biomolecule, that is carbohydrate, carbohydrate, protein, lipid, carbohydrate, protein, lipid, nucleic acid, etc. Then we can also classify the biomolecules into two groups depending on their molecular weight. Depending on the molecular weight of the biomolecules, we can classify the biomolecule into two groups. These are first one is micromolecule, and macromolecule, Which are micromolecule? Micromolecule means if the molecular weight in between 18 to 800 Dalton or less than 1000 Dalton. Micromolecules are those molecules if the molecular weight is less than 1000 Dalton, such as example, water, gases, amino acid, sugar, etc. These are the micromolecule. And what are the macromolecule? If the molecular weight is greater than 1000 Dalton. Macromolecule are those which have the molecular weight is greater than 1000 Dalton. Okay, such as example, carbohydrate, protein, lipid, etc. These are the macromolecule which are present in a cellular pool. Fine. Now we discuss about the percentage of different biomolecules in a cell and in a human beings. So I make a list that constituents percentage in a cell, percentage in a human. constituents in a cell, percentage in a cell in a human. Then first, water. Water is the main constituent of a cell and also a human beings or organism. So constituent is water. Percentage in a cell, it 70 to 90% of a cell. In human beings, the percentage of water is near about 61.6%. Next, the second one is protein, because protein is the structural protein. The percentage of protein in a cell is 10 to 15%, but percentage in a human beings it will be 17%. Then third, lipid. Lipid present in cell near about 2%, 2%, but inside a human beings lipid may be 13.8%. Okay. Then fourth is carbohydrate. Carbohydrate. The percentage of carbohydrate in a cell is near about 3%, but in a human beings it will be 1.5%. Then the minerals. Minerals is near about 5 to 8% of a cell, but in human beings it will be 6.1%. So these are the amount in percentage of different constituents or biomolecule in a cell and in a human beings. First I discuss about carbohydrate. Carbohydrate. You must remember that carbohydrate is the most abundant dietary carbohydrate are the most abundant organic molecule in nature. Remember it. Carbohydrate are the most abundant organic molecule in nature. Most abundant organic molecule in nature is carbohydrate. Okay. Carbohydrates are also defined as the organic substances having carbon, hydrogen and oxygen, where the ratio of hydrogen and oxygen is 2:1. So carbohydrate is made up of carbon, hydrogen and oxygen, and the ratio of hydrogen and oxygen is 2:1. You must also remember that the carbohydrate are the most abundant dietary source of energy. Means the energy which are need by us, and most of the energy supplied by the carbohydrate. Okay, and each gram of carbohydrate produces 4 calorie energy in organism. Okay, so the calorific value, okay, so we can uh, write that the the most abundant dietary source of energy the most abundant dietary source of energy is carbohydrate is carbohydrate. And the calorific value is 4 calorie per gram 4 calorie per gram. Okay, now we can classify the carbohydrate into, then we can classify the carbohydrate into following three groups. The groups are first monosaccharide, oligosaccharide, oligosaccharide and polysaccharide. Monosaccharide, we can classify the monosaccharide depending on different way. We can classify the monosaccharide in different way into different group, such as first in presence of the first and we can classify the monosaccharide depending on the depending on functional group. Depending on functional group, we can classify the monosaccharide into two groups. These are aldose sugar and ketose. Aldose means if the functional group in a monosaccharide is aldehyde, means CHO. If the functional group is aldehyde, then the monosaccharide also called aldose. If the group is keto, means C double bond O, then it is called ketose. As example, glucose. Ribose. These are the aldose sugar. In other hand, fructose, ribulose are the ketose sugar. Okay. Then we also can classify the monosaccharide depending on the number of carbon atom. Depending on the number of carbon atom. Depending on the number of carbon atom, we can classify the monosaccharide into following groups. That is, these are the triose sugar. These are the triose sugar, tetrose sugar, tetrose sugar, pentose sugar, pentose sugar, hexose sugar, hexose sugar, and heptose sugar. Hexose sugar and heptose sugar means the number of carbon atom in triose 3, tetrose the number of carbon atom is 4, pentose is 5, hexose is 6, and heptose is 7. Example of triose sugar: glyceraldehyde, glyceraldehyde. Example of triose sugar is glyceraldehyde. Tetrose, erythrose. Erythrose is the example of tetrose sugar. Pentose sugar, ribose. Ribose sugar is pentose. Hexose, you all knows that glucose, fructose, heptose, pseudoheptulose. Pseudoheptulose. These are the heptose sugar. Okay, so we can classify the monosaccharide depending on the number of carbon atom. The groups are triose, example glyceraldehyde; tetrose, example erythrose; pentose, example ribose; hexose, example glucose, fructose; heptose, pseudoheptulose. Then oligosaccharide. Oligosaccharides are the small carbohydrates. Oligosaccharides are the small carbohydrates which are formed by condensation of 2 to 9 monosaccharide. Here the monosaccharide are attached through glycosidic bond. Okay, so oligosaccharides are composed of 2 to 9 monosaccharides. 2 to 9 monosaccharides. Example: sucrose. Sucrose is also called disaccharide because sucrose is formed by the attachment of one molecule of glucose and one molecule of fructose. Okay, for the presence of one molecule of glucose and one molecule of fructose, for that sucrose also called disaccharide. Maltose also called disaccharide because it contains two glucose molecule. Then galactose also a disaccharide because it composed of one molecule of glucose and one molecule of lactose. Okay, without those, here raffinose, stachyose are the oligosaccharides. Then polysaccharide. What is polysaccharide? Polysaccharide consists of repeating units of monosaccharide or their derivatives held together by glycosidic bond. And polysaccharides are composed of more than 10 monosaccharide. Okay, polysaccharide is composed of more than 10 monosaccharide. The monosaccharide in a polysaccharide and the monosaccharides in a polysaccharide are attached by the glycosidic bond. Okay, then We can classify the polysaccharide into two groups. These are the homopolysaccharide. These are the homopolysaccharide, and heteropolysaccharide. Homopolysaccharide means if the polysaccharide is composed of only one kind of monosaccharide, then it is called homopolysaccharide. As example, starch, cellulose. As example, starch, cellulose are the homopolysaccharide, because if we break down the starch, then we can get many glucose molecule, only glucose molecule. If we break down the cellulose, then we also can get only glucose molecule. For that, these are the homopolysaccharide. And heteropolysaccharide, if different kinds of monosaccharide participate to form a polysaccharide is called heteropolysaccharide. As example, hemicellulose, chitin. These are the heteropolysaccharide. So that is the classification of carbohydrate. Okay. Then the derivatives of monosaccharide. Derivatives of monosaccharide. Monosaccharide are modified variously to form a number of different substances. Monosaccharides are modified variously to form a number of different substances. Those substances are first, deoxy sugar. Deoxy sugar means absence of oxygen. As example, deoxyribose, which are the derivative of ribose. Okay. In second carbon of the deoxyribose, it contains only hydrogen, not hydroxyl group. And in case of ribose sugar, the second number carbon atom holds a hydrogen and a hydroxyl ion, but in case of deoxyribose sugar, it contains two hydrogen ion. Okay, for that, the deoxyribose sugar is the example of deoxy sugar. Then amino sugar. Amino sugar means if any hydroxyl group replaced by amino group, then it called amino sugar. As example, glucosamine. N-acetyl glucosamine, N-acetyl muramic acid are the main composition of the peptidoglycan bacterial cell wall. So those are the amino sugar. Then the sugar acid. Then the sugar acid. Sugar acid means if any glucose, if any monosaccharide contains the carboxyl group. If any monosaccharide contains the carboxyl group, then the monosaccharide is called sugar acid. As example, ascorbic acid. As example, ascorbic acid is a sugar acid. Ascorbic acid is a sugar acid. Glucuronic acid. Glucuronic acid is also an example of a sugar acid. Then the sugar alcohol. Then the sugar alcohol. Sugar alcohol. They are produced by the reduction of aldose or ketose. They are produced by reduction of aldose and ketose. Okay. As example, glycerol. As example, glycerol, which is the main composition of lipid. Which is the main composition of lipid, or which participate or takes part in lipid synthesis. Another is mannitol. Mannitol. There is mannitol. It present in some fruit and some brown alga also. And some brown alga also. So these are the classification of carbohydrates. Now I discuss some technical term such as stereoisomerism. What is stereoisomerism? Stereoisomerism is an important character of monosaccharide. Stereomerism is an important character of monosaccharide. Stereoisomers are the compounds that have the same structural formula but differ in their spatial configuration. Stereoisomers are the compounds that have the same structural formula but differ in their spatial configuration. As example, okay, first I write this stereoisomerism. Stereoisomers are the compounds that have the same structure, okay, but differ in their spatial configuration. As example, which that is the structure of 3 carbon compound, means glyceraldehyde. Okay, the stereoisomers is also that the mirror image, that C H H O CH2 OH. This forms both are the stereoisomer. Okay, these are the glyceraldehyde. Look, here is the OH group, here is the OH group, stereoisomer. Then another term is optical activity of sugar. Optical activity of sugar. Optical activity is a characteristic feature of compounds with asymmetric carbon atom. So, optical activity is a characteristic feature of compound with asymmetric carbon atom. In carbohydrate, we can see the asymmetric carbon atom is present. For that, they shows the optical activity. If a beam of polarized light is passed through a solution of an optical isomer, it will be rotated either to the right side or left side. If a polarized light is passed is passed through a solution of an optical isomer it will be rotate to either right or left side. If it rotates the light beam, if the light beam rotates to the right side, then it called dextrarotatory. If rotates to the left is called levorotatory. Okay, dextrarotatory also called D form, and levorotatory also called L form. As example, I can draw the linear structure of a glucose. It has two forms, that C double bond O H means CHO, then COH, H, C, H, HO, then COH, H, 1, 2, 3, 4, COH, and H, CH2OH. Okay. Another form is CHO, then CHHO. It is also the mirror image. COH, H, CH, OH, CH, OH, and CH2OH. 1, 2, 3, 4, 5, 6. Fine. Look, here the OH present in the right side, so it is the D-glucose. So it is the D-glucose, because if we can pass the polarized light from this solution, then the light will be rotated to the right, and here the OH group present in the left side, for that it called L-glucose, because it rotates the polarized light into left. Why it does occur? Because for the presence of asymmetric carbon atom, and this is the optical activity. Okay. Then epimers. What is epimer? If two monosaccharides differ from each other, in their configuration, around a single specific carbon, they are referred to as epimer to each other. Okay. Then we can write, if two monosaccharide saccharides differ by their configuration around a single specific carbon atom. Then those monosaccharides are called epimers to each other. As example, glucose and galactose. Are the epimers. Regarded to the carbon 4. As example, I can again draw the linear structure of glucose. Here 1, 2, 3, 4, 5, and 6. CHO, OH, H, OH, OH, CH2, OH. Then there H H HO H. That is the structure of glucose. 1 2 3 4. So if we observe the configuration of 4C of 4C is differ in galactose. In galactose, CHO aldehyde group 2, 3, 4, 5 and 6, CH2 OH. Then here OH, all are same, only single carbon atom are differ, is different, single carbon is different. HO here, the H here, HO, then OH and H. Look at the number 4 carbon atom. Only this configuration is different. Only the 4 carbon configuration of carbon atom, fourth carbon atom are different. Then it is the structure of glucose, and it is the structure of galactose. Structure of galactose. So galactose is the epimer of glucose, and glucose also the epimer of galactose. Okay. Then reducing sugar, reducing and non-reducing sugar. Non-reducing sugar. What is reducing sugar? What is reducing sugar? Reducing sugar has, the reducing sugar has a free, has a free aldehyde group, means CHO, or a free keto group, has a free aldehyde group or a free keto group. Then it converts, the cupric ion, then it converts the cupric ion, means Cu2 plus, to cuprous ion, Cu plus, cupric ion to cuprous ion. Okay. So reducing sugar are those sugar which has a free aldehyde group or keto group. If they do not have the free aldehyde or keto group, then they does not convert the cupric ion to cuprous ion. Those sugars are called non-reducing sugar. Okay. So reducing sugar don't have a free aldehyde group or a keto group. Oh, don't have a aldehyde group or keto group. For that they does not convert. It does not converts cupric ion to cuprous ion. As example, glucose, fructose are the reducing sugar. Glucose, fructose are the reducing sugar. But as example, sucrose is a non-reducing sugar. You all know that the sucrose is formed by one molecule of glucose and 1 molecule of fructose. Then why does the sucrose is a non-reducing sugar? Because at the time of attachment of glucose and fructose, they lose their free aldehyde group and keto group also. Okay, for that sucrose is the example of a non-reducing sugar. Is a example of a non-reducing sugar. Then pyranose and furanose. Pyranose and furanose. Pyranose and furanose. What is pyranose? If we make a solution, okay, if we make a solution of a glucose, sorry, of a monosaccharide, in that solution, they can form a ring. They form a ring in a solution. If the ring is, if the ring will hexagonal, if the ring will hexagonal, if the ring will hexagonal, then it is called pyranose. If the ring will pentagonal, then it called furanose. If the ring will hexagonal, that is O, they attach by the OH group. O. If the ring will hexagonal, then called pyranose. If it form a pentagonal structure, then it's called furanose. Okay. When pyranose will form, if the first carbon atom, attach, if the first carbon atom attach with the fifth carbon atom, then they form first carbon atom attach with the fifth carbon atom by oxygen, then it formed a hexagonal structure called pyranose. If the first carbon atom attach with the fourth carbon atom by oxygen. First carbon atom attach with the fourth carbon, then they form the pentagonal structure. If the first carbon attach with the fifth carbon, then it form the hexagonal structure. So hexagonal structure is called pyranose and the pentagonal structure is called furanose. Thank you.